Okay, we will continue chapter 6 today. Okay, so we will discuss the activation energy and enzymes. Now, what is activation energy? So just remember on the previous lecture, we talked about two types of reaction, which is exergonic and endergonic reaction. Okay, exergonic reaction is also known as catabolic reaction, while the endergonic reaction is known as anabolic reaction. So I will show you this exergonic reaction first, okay, the catabolic reaction. As we see over here, even though the exergonic reaction or catabolic reaction is spontaneous, it means that it can go spontaneously. However, it will not do by itself. If you see this picture over here, you see this the harms. Okay, so again, it's different color. Okay, so the harm over here, this is what we call the activation energy. Okay, so these reactants, which is bigger molecules, will not go right away to produce the product without going to the or needing the activation energy. So this energy is go up over here. So it requires something in order to perform these reactions. Again, the energy that need to activate this reaction is called the activation energy. So therefore, the activation energy can be defined as the energy required for a reaction to proceed. Okay, very easy examples. Okay, very easy example for this exergonic reaction. So when we put gasoline in our car, now our car full with the gasoline, do you think the car will go by itself, will run right away? No, because what? Because we need to activate the reaction. So we need to start the car. When we start the car, what happened? Actually, the spark in the engines will activate the reaction. So after the spark activate the reaction, then the potential energy from the fuel, okay, can be converted into the kinetic energy of the car. So the car now can run or can move. So this is what we call the activation energy. Every energy, every reaction will have this activation energy. Another examples, we know that in the log, for example, in the coal, there are a lot of potential energy, but it will not burn by itself. We have to give something. Okay, we have to get match, and then we start burning the coal or the wood. What happens whenever it starts? Then the coal or the wood will be burned until finished. So the reaction will go after we give something, which is what we call the activation energy. So activation energy, again, is the energy required to start reaction. Reaction will not go by itself without the activation energy. The same thing inside our cell, yeah, inside our cell, inside our body. So all reaction require activation energy. It will not do by itself without giving the activation energy. So most of the time, the heat that we have in our body. That's the reason why we have a body temperature at least maybe 96 to 98 degree Fahrenheit. And this heat can spark the reaction. The highest temperature, the faster the reaction will start. Now, our body cannot be more than, let's say, 100 or 99 degree, okay, or 100. If our body temperature increase above 100, what happens? We have a problem. Yeah, we have an illness. We cannot do that. So it means that the heat in our body that's about 96 to 98 Fahrenheit degree is actually not enough. Okay, not enough to start reaction. Okay, so we need something else in order to start the reaction. So the one that we need in addition of heat is called the catalyst. Okay, so we will have some things or molecules in our body, in our cell, that we produce to increase the activation energy, right? In addition of heat that we have. Okay, so that's the things that we need, the catalyst. And we produce a lot of catalyst in our body. Okay, so catalyst, catalyst, what is catalyst? Catalyst is molecules. Is molecules inside our cell or inside our body that speed reaction by lowering the activation energy. Look at this picture over here. You see this in the picture? Okay, you see the blue line over here? Okay, this is the activation energy without catalyze. It's so high. Okay, we need a lot of energy. But if we do have catalyze, if we have catalyst over here, the activation energy is become lower. Look at this one, it's lower. Okay, so this catalyst is actually will speed the reaction by lowering, by lowering the activation energy. So that's the definition of catalyst. Again, catalyst is a molecules that speed reaction by lowering the activation energy. When the activation energy is lower, what happens? Then the reaction will start faster. Okay? So these reactants will be converted into the product faster, compared to without the catalyst. And most of the time, if we do not have catalysts in our body, in our cell, this reaction actually will not start because the activation energy is too high. It means that this catalyst is really needed. So this catalyst is most of the time is required inside the cell, to start metabolism, whether catabolism or anabolism. So it is required. So we need catalyst. And the catalyst in our cell is known as enzyme. So enzyme is a catalyst. An enzyme, 99% enzyme is actually proteins. So enzymes are proteins that has a function as catalyst, which is molecules that speed reaction, by lowering activation energy. Okay, again, from this definition, then we know that enzyme is required. We have to have enzyme in order to have reaction inside our body. If we don't have it, then we will have abnormality, maybe sickness or illness. For example, is people that do not produce enzyme called lactase, then they will have problem when they drink or when they take lactose or milk as their diet. Because what? Because that lactose will not be converted into galactose and glucose without the enzymes called lactase. So again, okay, enzyme are required in order to start reaction inside our body. Okay, so again, this enzyme, they are 99% protein. There are some enzyme that is not protein, but most enzyme, okay, majority of enzymes are proteins that catalyst, which is speed reaction by lowering the activation energy. Enzyme will bind to the reactant or the substrate molecule, and then it will make a process to produce the product. So enzyme is actually, okay, the catalyst that will bind to the substrate, and then after that, it will produce something. It will produce the product. If you see here in the catabolic reaction, for example, okay, so both catabolic and anabolic reaction, they require enzyme. For this catabolic reaction, we already mentioned that without the enzyme, the reaction will never happening. Because what? Because the energy activation is really high. Okay, so in order to lower that, then we need enzyme. So when the enzyme present, this reaction will start faster, and will start. Without enzyme, the reaction will never start. The same thing with the anabolism. Remember, this is synthesis reaction. Start with the reactant that have lower energy to make a product that have lower energy. It means that it requires a lot of energy. Okay? So without an enzyme, the energy that's needed is very high, up over here. Okay? Sometimes this reaction will never happen because the energy is too much. We need too many energy in order to process this. With the enzyme, this reaction will be faster or will be happening because this activation energy will be lower over here. Okay? So this reaction, anabolic reaction, also require enzyme. So again, enzyme is a very important molecules in our cell, in our body, in order to start reaction. They will increase the speed of the reaction by lowering the activation energy. Enzymes are very specific. Again, they are protein, and they are very specific to specific reaction. They usually, the name usually based on the name of substrate or reactants that they will change. For example, okay, there are these are the name of some enzyme. Okay? Usually enzyme name end with ase. So this is what we call the suffix. Okay, the suffix name for an enzyme always end, most of the time end with ase. Like for example, protease. So what is this? This is enzyme that actually will break down protein. Okay, so protease, so protein. How about lipase? This is the enzyme that break down the lipids. Lactase. This is the enzyme that break down the lactose. Okay, so the name of the substrate usually end with ose. And then sucrase, this is the one that break down the sucrose. Maltase, this is the enzyme that break down the maltose. Okay. Amylase, this is the one that break down the amylum or starch. Okay, break down the starch. Peptidase, this is the one that break down the peptide, which is another type of protein. DNAase, this is the one that break down DNA into nucleotides. RNAase, this is the enzyme that break down the RNA into also nucleotides. Helicase, this is the enzyme that break down the double helix, the DNA. Okay, the DNA have a double helix molecule like this. When helicase present, it will make like this. Okay, just like a stairs. Okay, like a ladder. Okay, it's not. It's open there. The helix molecules. Endonuclease. This is the enzymes in the DNA that fix problem in the DNA. DNA polymerase, and the name is telling you, this is the enzyme that actually will form the DNA polymer. So this is, over here is going to be anabolic, okay, anabolism, which is making bigger molecule. Over here, mostly this is catabolism, okay, catabolic reaction over here, okay. RNA polymerase, this is the one that will make RNA. Lysozyme, we already mentioned lysozyme. This is the one that cut the foreign molecules like bacteria. Sometimes the name of enzyme also end with IN. Trypsin, chymotrypsin, pepsin, pepsinogen. Mostly, if it is like this is also the one that work. The substrate is actually protein. So these are some examples, the names of the enzyme. They have a very specific names because they are also work for specific substrate. Now the structure of an enzyme. Okay, the enzyme is look like this. Okay, this is actually like a 3D molecules. Okay, that's the structure of enzyme, which is a 3D molecules of proteins. So if we can review the protein structure, okay, you still remember there are four protein structure. The first one is called the first or primary structure. Okay, so it's look like the primary structure of proteins is actually just looking at the structure or the bond between amino acid. So this is amino acid, amino acid, amino acid. Okay, so this is amino acid. We just look at this connection, just bond between amino acid. That bond is called the peptide bond. That's the primary structure. The second one is called the secondary structure. Okay. Secondary structure look like this. So we look at the primary connection. Okay. Let's say like this. Okay, that's the primary structure, right? But in addition of that, there is also connection with the hydrogen bond that make this protein become making this kind of helix structure. So this is the helix. Okay? This is the secondary structure. In addition to look at the peptide bond, there is another bond that make this protein change the structure. Sometimes it's gonna be look like this. So you have protein, amino acid over here, amino acid, amino acid, amino acid, amino acid, and between this amino acid, there will be a hydrogen bond. Okay, this is what we call the pleated sheets of the protein. So these are the secondary structure. And then we have the third one, tertiary structure. Okay, so in addition of this connection between peptide, okay, amino acid, and then also the hydrogen bond, there is another covalent bond that present between the amino acid and causing this protein become 3D molecules. Okay, so it's kind of ball structure, globular structure. This is actually mainly the structure of the enzyme. So enzyme is a tertiary structure of protein. Okay, and then we know the last type of proteins structure is called the quaternary structure. Okay, so this is one tertiary with another tertiary, another tertiary, another tertiary. They combine together. Let's say hemoglobin, for example. Hemoglobin actually, it has 4 tertiary structure that is bind together with irons in the middle. Okay, this gives the quaternary structure. But we are focused in this tertiary structure over here, which is the structure of enzyme. So all enzyme, they are tertiary structure of the proteins. So because of that, if you see the ball, sometimes they have area or site. So this tertiary structure give the enzymes the ability to make or to create a site. There are 2 types of sites in an enzyme. The first one is called the active site. Okay, so active site is the site where the substrate bind. Okay, so this is the place for the substrate bind. This is what we call the active site. The other site is called the allosteric site. This is the site where other things, other molecules, okay, maybe inhibitors or maybe activator bind. Okay, this is the site when other molecule will bind. Okay, so again, there are 2 main sites in enzyme. The first one is called the active site. This is for the substrate to bind. And the allosteric site. This is for other molecules to bind. Sometimes these molecules are also needed as a activator, for example, to activate the enzyme. Now, how the enzymes reactions look like. And remember, there are active site. The active site is the most important site for the enzyme to work. Because what? Because that's the site when the substrate will bind. Okay, so the active site, this is the site where the substrate will bind. When the substrate binds, it will give the fit, it's called the induced fit, or sometimes, mostly, it used to be called as a lock and key. Just like, when you enter your house, you have the key. It has to fit with the lock in your door. Otherwise, it will not open. So this is the same idea. The substrate must be fit with the active site. of the enzyme. Okay, so when the substrate fit with the enzyme, then it will, so this is the enzyme, it will bind to it. When it is bind, that what happened, it will create the molecule, is called the ES complex, enzyme substrate complex. Okay, it is required time over here. There is something happening, whether it will break down or maybe it will synthesis. And then after that, it will go to the next molecules. This is intermediate molecule. It's called the enzyme product complex. Okay, so both of these is called the intermediate molecules. The ES complex and the EP complex, they are intermediate molecules between substrate and the product. And then after the EP enzyme product complex, then the enzyme will release the products. Okay, enzyme will release the product. At the end, the enzyme will separate with the products. So maybe this reaction is a catabolic reaction from one big molecule into two smaller molecules over here as a product. So this enzyme, after being used, it can be used again. So will be used for the next reaction. Okay, for the next reaction. Okay, so enzyme is actually never gone from the reaction. It will be used again, against, and against. That's the reason why we only need a small amount of enzyme. We usually create small amount of enzyme because what? Because it can be used again and again. So this is how the enzyme work. The enzyme can be regulated. Okay, this is the important things over here, because without that, then the enzyme will always work. So they will be regulated. Okay, so most the regulations is happening automatically inside our body. Okay, for examples, when we eat, and the food will go to our stomach, for example. When the food present in our stomach, then the digestive cell, okay, which is will produce a lot of enzyme, will start the reactions. Without the food in the stomach, then the enzyme will not be working. So they will be relaxed. So this is really important actually, okay? When we start to sleep, want to sleep, so make sure you don't eat a lot of meal or don't eat before sleep, because what? If we eat before sleep, our digestive cell, our stomach, for example, will continue to work because they don't actually, the stomach will not really care whether we are asleep or not. The most thing that they care is if there is food inside the stomach, then the stomach will digest that food, whether we are sleeping or we are awake. Okay, so this is one of the regulation. Another regulation is the temperature and pH. This is also can increase or decrease the reaction. Most of the time, the temperature will increase, okay, or start the enzymatic reaction. There is another thing that also regulates the enzyme. It's called the promoter or activator, or inhibitor, allosteric inhibitor. Okay, so there are two types of dysregulation. The first one is called the allosteric activator, which is the molecule that modify the active site to activate the enzyme. So for example, remember, let's say the enzyme look like this. Okay, let's say it has active site. Okay, and this is allosteric site. Okay, let's say, okay, before it is activated, okay, gonna make it over here. Let's say we're gonna top over here. Let's say before it is activated, it look like this, and the substrate is look like this. So this is the substrate. Okay, so if you see over here, this one will not happening. The reaction will not happening because what? Because the substrate is not fit with the active site, right? So when when the activator enter into the allosteric site over here, this change the active site become like this. So this reaction will be happening. So this one over here is called the activator. Okay, because this activator bind to the allosteric site, this site over here, then this activator is called the allosteric activator. Okay, allosteric activator. Okay, so easy to understand, right? Allosteric activator is the activator, the molecules that activate the enzyme by changing the active site into become fit with the substrate. Now, how about the allosteric inhibitor? This is similar. Okay, let's say this is the substrate like this. Okay, and then this is the active site. Okay, so without this inhibitor, this reaction will be happening. Okay, but when the molecule bind to this, okay, the molecule bind to the allosteric site over here, the active site is changed. Okay? So the reaction will not happen. So these molecules over here is called the inhibitor. It will inhibit the reaction, and because it bind to the allosteric site, this inhibitor is called the allosteric inhibitor. Okay, so this is just opposite. Okay, for the activators, usually start with the active site that is not fit with the substrate, and then we need the activator in order to make it fit. And then if it is inhibitor, usually the enzyme is actually already fit with the substrate, so the active site already fit with the substrate, and then this molecule bind to the allosteric site. It's causing the enzyme will not fit again to the substrate. It will inhibit the reaction. So I hope it's easy to understand the difference between activator and inhibitors. Now again, for the activator, allosteric activator, usually there are two types. The first one is called the cofactor. This is when the activator is actually inorganic molecules, such as ions or metals, okay, magnesium, zinc, irons, for example. They are inorganic molecules. This is simple molecules, remember. And if it is organic molecules, okay, or complex molecules, bigger molecules, the activator is called a coenzyme. Okay, let's say if it is an enzyme like this is the substrate. Okay, and then if this activator bind to this, if it is inorganic, then it will open active site like this. Okay, so this inorganic molecule over here that bind over here is called a cofactor. cofactors. Okay, but if the one that bind to the allosteric side is actually bigger molecule, okay, maybe like this. Okay, so this one is called coenzyme. Okay, so the difference is actually the type of molecules. If it is inorganic molecules, that activator is called cofactor. If it is organic molecules, like vitamin, for example, the activator is called the coenzyme. So again, this is the reason why we have a lot of inorganic molecules like iron, zinc, for example. They are very important for us because many of them is actually cofactor for an enzyme. And vitamins like vitamin A, B, C, D, E, K, for example, they are really important because many of them is actually part of the enzyme is called the coenzyme that will activate the enzyme. Without them, our enzyme will not be active, will not be able to process the reaction. Okay, so this is really important additional molecules that make the enzyme work, whether it is cofactor or coenzyme. There is another also, the type of inhibition. Okay? So if it is inhibitors, there are two types of inhibitors. First one is called the competitive inhibitors. The second one is called the non-competitive inhibitors. What is competitive mean? So competitive inhibitor is a molecules that compete for the active site with the substrate. So this inhibitor is actually compete with the substrate for the active site. So let's say this is an enzyme like this, okay? And then let's say this is a substrate, okay? Let's say this is substrate like that, which is fit with the active site. Okay, and then there will be another molecules that very similar with the substrate. So this is inhibitor, because this inhibitor compete with the substrate. So this one substrate, right? Substrate. So this one compete for active site. When they compete for the active site, then what happens? The reaction will be slowing down. Okay, so if you see this one, this is a normal reaction. This is the normal reaction over here without inhibition. Okay, if there is a competitive inhibitors, the reaction will be slowing down. But if we increase the substrate, so we add more substrate, we eat more, for example, or whatever we do, we add more substrate, what happens? The reaction will go back to the maximum rate. Yeah, it's going back to the maximum rate over here. Okay, so this is maximum rate. So in this graph, it shows you that the competitive inhibitors will slow down the speed of reaction. However, it will not change the maximum speed. So the maximum speed finally will be reached again over here. Okay? It will be reached again. If this is the competitive inhibition. Now, how about the non-competitive inhibitors or inhibition? This is when the let's say this is substrate. Okay, so this is fit over here. And then when there is a Okay, this is substrate, this is substrate, and then this is the inhibitor. Inhibitor. This is substrate. Okay, so if you see over here, the inhibitor is actually does not compete with the substrate, because this inhibitor actually bind to the allosteric site, not to the active site. Okay, so whenever this inhibitor bind to the allosteric site, it will change this active site. It will not fit again for the substrate. So it does not matter whether you add more substrate over here, right? You add 1,000 of substrate over here, it will not increase the reaction. And the reaction will never reach the maximum speed. Okay, so you see over here, the reaction will never reach the maximum speed over here. Okay, so this is the act of non-competitive inhibitors. It will bind to the allosteric site and it will slow down the reaction. At the same time, it will also reduce or decrease the maximum speed. So both of these will be affected by the non-competitive inhibitors or inhibition. So both the reaction and the maximum speed will be reduced. But for the competitive, on the reaction speed reduced, but the maximum at the end will be the same. Okay, so I hope this easy for you to understand the difference between competitive inhibitors and non-competitive inhibitors. Now, there is another thing. This is the last thing for this enzyme metabolisms. Okay, so most of the time in our body, the reaction is not one step. Okay, from substrate to, like, from a substrate, okay, with enzyme, for example, then become product. Let's say enzyme over here. Substrate, enzyme, product. No, most of the time, okay, when it is substrate, it's become first product, second product, third product, fourth product, until finally it has, for example, final products. Okay? And each reaction over here requires enzyme E1, E2, E3, E4, E5, for example. So this reaction over here, the common reaction that happening in our body using the enzyme is called the metabolic pathway. It's not only one way, but it will have many steps and require many enzymes. So the pathway that require many enzymes is called the metabolic pathway. Requires many enzymes to produce the final products. Okay, so this is what we call the metabolic pathway, which is the pathway that require many enzymes to produce the final products. Now, when the final products produce, when it is reaching the maximum concentration, okay, let's say we only need 1,000 products, final product. When it is reaching the 1,000 products, and then it's go maybe to 1,001, 1,002, So when it's reached the maximum number, then these products will become inhibitor, mostly for the first enzyme. Okay, it's become inhibitor. Okay, for uh, the first enzyme or the second enzyme. Okay, so this inhibition over here is known as the feedback inhibition. Okay, so inhibitions that is caused or done by the final product in order to stop the products. So we don't want to make a lot of products. It's enough. In order to stop that, then the final product become inhibitor for the first or the second enzyme. Okay, so this is what we call the feedback inhibition. The inhibition that is done by the final product in order to create the normal amount of the final products. So it will not be excess of products. I think that's all for this chapter. So we're done with chapter 6. So make sure you study again chapter 6, part 1, and then this part 2, and then use them to finish your assignment for chapter 6. And then forget also there will be quiz for this chapter, metabolism, on the next class meeting. Goodbye. Thank you very much.